Good evening, Pratham. Okay, so I'm Rahul Saha, and in the last class we had discussed about the specificities of the trios transporter, okay, the PI antiporter also, like how does it regenerate NADH and how does it cause the transportation of NADH from the intermediate space to the, you can say, matrix. And also I had described the details of the C2 cycle. The C2 cycle is also known as the photorespiration cycle. Okay, in some books it is also given as photorespiration. Okay, it causes the, you can say, use of or net usage of, you can say, 2 NADH or 2 NAD plus are used up, or sorry, 2 NADH are used up to form 2 NAD plus. Okay, and here 1 NAD plus will be used up to form NAD plus in the hydroxypyruvate 2 glycerate cycle, but 1 NADH is regenerated also, okay, when glycine is oxidized to serine, okay, via glycine decarboxylase. But in the end, what you do is we have to use one ATP to generate ADP and PI in order to cause the phosphorylation of glycerate to form 3-phosphoglycerate, right? So that we have to do. Then this takes part in 3 cellular organelles. You have the chloroplast, then from where the 2-phosphoglycolate becomes glycolate and gets transported to the peroxisome, and then via various reactions the glycolate becomes glyoxylate, then glycine comes into the mitochondrion, the third organelle, and then becomes serine and goes back into the peroxisome. Then it goes back into the, again, chloroplast, where it is converted into 3-phosphoglycerate, okay, the form glycerate that goes in. Okay, so that is the reason behind the production of C4 cycle, okay, or the evolution of C4 cycle in C4 plants, okay, that in order to this, you can say, eradicate this photorespiration or the C2 cycle, the C4 plants have evolved. Okay. Now, how does it occur? Now, in many plants that grow in the tropics, like in the temperate zone, crop plants native to the tropics, such as maize, you can say sorghum, you can say sugarcane, a specific or a very unique, a special mechanism has evolved to circumvent the same problem of photorespiration or the perpetuation of C2 cycle. They cannot afford, they can ill afford to let the C2 cycle being perpetuated. Okay. So the thing is, the step in which CO2 is fixed into a 3 carbon product, which is your 3 phosphoglycerate, that step is the most important step. If you can make your way through or try to, you can say, modify that step, not that step directly, you can proceed or you can say make some additional changes in the preceding steps which lead to the formation of 3 phosphoglycerate. Then if we apply enough common sense, we can evolve a better pathway, okay, so that we can directly eliminate the C2 cycle. Means elimination of C2 cycle means what? We need to, there is a need to eliminate C2 cycle or photorespiration. Photorespiration. Now, why would the photorespiration be need to be eliminated in case of C2 plants? Because C2 plants, they are not getting enough, you can say, sunlight all the time. So whatever sunlight they're getting, they need to make proper use of it and need to maximize the production of starch. Otherwise, the NADPS that has been produced, the ATPs that have been produced via photosynthesis would be wasted because it's a wasteful process, it's a futile process that I've already explained to you, the photorespiration. So they can ill afford to go for this kind of photorespiration. Okay. So the thing is, what you need to do is you need to take out, you need to eliminate this pathway. So in order to eliminate this pathway, what are you trying to do? You are trying to take out the effective catalytic pathway pertaining to the oxygenase activity of the enzyme Rubisco. So ribulose 1,5 bisphosphate, okay, oxygenase carboxylase. So or carboxylase oxygenase. So you need to take out, it has dual activity, carboxylase oxygenase. Okay. High Km, low Km that we have talked about. Okay, this one low, this one high. But still 3:1 it is going for O2 addition with ribulose 1,5 bisphosphate to produce 3 phosphoglycerate and 2 phosphoglycolate, 5 carbon total, right? So we need to eliminate the formation of 2-phosphoglycolate. This is a 2-carbon compound which is a completely futile product, a wasteful product which needs to be again recycled because these 2 carbons need to be salvaged. Otherwise, you cannot regenerate RUBP. Otherwise, you wouldn't need to salvage it also. Okay, you would ask the question, why do we need to salvage it? Let it go, let it be, let it go to, you can say, the metabolic, you can say, catabolic pathway and be, and then the various products can be regenerated, the various atoms we can get. But the thing is, if you do not regenerate, I told you 3 into 3, the 3 RUBP plus 3 CO2 are added to produce 6 phosphoglycerates, out of which 5 into 15, 5 into 3, because each of the 3 phosphoglycerates are of 3 carbon compounds, they are used up to regenerate the Rubisco, right? If you do not regenerate Rubisco, the Rubisco concentration will fall, will plummet, and then you cannot even ponder upon or imagine the catalysis of the starch synthesis, and then the plant would wither and die at night. Okay, So you need to salvage it because of that very reason. Now the salvage pathway is a completely wasteful process. It requires an ADH, requires your ATP. So that you can ill afford in case of C4 plants, right? Maize, sugarcane, sorghum and all. Now what you are effectively trying to do is, what the evolution has effectively tried is to eliminate this oxygenase activity of ribulose 1,5 bisphosphate carboxylase oxygenase slash rubisco enzyme. Okay, which is the, you can say, most predominant enzyme in the entire biosphere as far as the concentration is concerned. You are trying to eliminate the oxygenous activity, but you are not trying, the nature has not changed the Rubisco gene so that its oxygenous activity is completely gone. Oxygenous activity is present, but it has gone for a roundabout manner, which is a very, very skillful manner and enthralling, amazing activity that the evolution has generated, so that Rubisco can never ever use its oxygenous activity even if it wants to. Okay, play with its km values and make sure it cannot use its oxygenase activity and in any circumstance. That is what has been done by, you can say, the C4 plants. Okay, so we know already that, once I told you that the preceding step has been modified. Okay, how it has been modified? So plants that use this process, okay, which have modified the preceding steps of the formation of 3-phosphoglycerate, are referred to as the C4 plants. Okay, and the assimilation process is known as C4 metabolism or C4 pathway. Why C4? Because the first compound produced there in C3 was 3-phosphoglycerate, 3-carbon compound. Here it is an oxaloacetate. So we have come across oxaloacetate many times in the Krebs cycle. So oxaloacetate is a 4-carbon compound. That is why the first compound here produced is an oxaloacetate. That is why it is termed as a C4 cycle. Now plants that use the carbon assimilation method, okay, that has been described, that I have described till now, use the first step of the reaction in which CO2 is catalyzed with ribulose 1,5 bisphosphate to form 3-phosphoglycerate, that we have already seen in C3 plants. But in C4 plants, what's really, really interesting and unique is they typically grow at very high light intensity and high temperature. Okay, and they does have several significant characteristics. Okay, the C4 plants, they grow at high light intensity and high temperatures. So they have the following specific and unique characteristics that make them very, very peculiar and demarcate them from the C3 plants. Okay, number one is high photosynthetic rate. High photosynthetic rate of photosynthesis is high. Okay, then high growth rates, producing more ATP and more NADH, so consequently the growth rate will be higher. High growth rate. Then you can see very low photorespiration. Okay, that I'll explain to you, that we are only discussing that how C4 will eliminate the photorespiration. So very, very low and negligible photorespiration, C2 cycle. So negligible photo respiration. Then what we have is very low rate of water loss and a specialized leaf structure. water loss and a specialized leaf structure. Okay, so these are the characteristics of the C4 plants. Now, photosynthesis in the leaves of C4 plants, it involves two cell types. Know it very, very well. One is the mesophyll cell, another is the bundle sheath cell. Okay, it involves two cell types. Very, very important question. Involves two cell types. One is the mesophyll cell, two is the bundle sheath cell. Bundle sheath cell. Okay. Now, there are three variants of C4 metabolism, okay, which was worked out in 1960s by Marshall Hatch and Roger Slack. They were the two scientists that worked out. We'll only describe one variant, okay. So in plants of tropical origin, okay, the first intermediate into which carbon is fixed is a four carbon compound oxaloacetate, the simplest bundle or the simplest way that it can be achieved. That is why it is known as a C4 cycle. Now this reaction, it occurs in the leaf of mesophyll cells. Okay, it occurs in the leaf of mesophyll cells, and the substrate is HCO3 minus, not CO2. Very, very important. I'll draw the reaction for you. The substrate is HCO3 minus, not CO2. Means CO2 is not reacting. Okay, what is reacting is HCO3 minus. Okay, and then oxaloacetate dust formed will be reduced later on to the malate and all. So that I'll show it to you. Or it can be converted to aspartate later on. Okay, it can be converted to aspartate also. Okay, so that can happen, but that reaction you may not be aware of. It is transamination reaction. Okay, so that is there. So that reaction we'll skip for now, the aspartate conversion. Okay, so what happens is, just look at this reaction that I'm trying to draw here. First of all, CO2 is in air. Okay, CO2 is in air. Now H2O becomes H plus. It is also in air, the water vapor. H plus. Okay. It becomes HCO3 minus, right? Now it has become HCO3 minus. Okay. HCO3 minus will react with glycolytic product. This is your mesophyll cell. Okay, it has a glycolytic product, phosphoenol pyruvate, from which pyruvate was produced, right? In the last reaction of glycolysis, the second substrate level phosphorylation, if you remember, phosphoenol pyruvate yielded pyruvate by the enzyme, catalyzed by the enzyme pyruvate kinase. Here you can use Mg2 plus or K plus or even plus as the coenzyme, right? Then we can say the tautomerism of pyruvate also happened in order to keto transformation. So that was there. Now this phosphoenol pyruvate is present in the mesophyll cell. This is the mesophyll cell. Okay. So now phosphoenol pyruvate SCO3 will enter. The enzyme will be PEP carboxylase, means carboxylation of PEP will happen. Okay, and then the phosphate will be released because it has got one phosphate group. That is how it could go for substrate level phosphorylation, because it donated the phosphate to ADP to produce ATP. Now that phosphate will be released, carboxylation will be done. So three carbon, this is a three carbon compound. Pyruvate becomes a three carbon compound, or is a three carbon compound in glycolysis. That's why. So now the PI will be released and then oxaloacetate will be produced. This is oxaloacetate. Okay, then oxaloacetate is produced. Now, after oxaloacetate has been produced, in Krebs cycle, if you remember, malate gets oxidized to oxaloacetate, right? When malate did get oxidized to oxaloacetate, what formed? This was the reaction. That's why all the cycles are interrelated in biochemistry. Okay. This was the last reaction of the Krebs cycle. This is an oxidation reaction. Okay, in case of Krebs cycle. So here, what did we get? We got something like oxidation, something must be reduced. NAD plus reduced to NADH. I got one more reducing equivalent. Enzyme will be dehydrogenase, as dehydrogenase falls under the category of the first category of enzyme known as oxidoreductase, right? So then I got the one more reducing equivalent that the Krebs cycle is meant for, to produce or provide reducing equivalents, right? Then I got NADH. From malate to oxaloacetate, we got NADH, right? Now, can we go for the reversal of this event? Very, the answer is a resounding yes. Why? Because dehydrogenase can catalyze both the reactions depending on the concentration. If malate concentration is more, it will forward it to oxaloacetate. If oxaloacetate concentration is more, it will reverse it to malate depending on NAD plus NADH availability. Or NADPH can also be used in case of plants. Nicotinamide adenine dinucleotide phosphate hydrogen. This is more used in plants, right? So depending on this availability and this availability, NADP plus, NADA plus, NADH, it can go for either of the two reactions or either of the two sides. This is essentially a reversible reaction, right? With a very less, or we can say negative del G also. Okay, but since oxaloacetate is taken out of the, you can say, system so quickly by reacting with acetyl CoA, does it is forwarded in the, you can say, or it proceeds in the forward direction, but the del G is negative for reaction of this side. Okay, so now what you can do is we can go for the opposite reaction, right? We can go for oxaloacetate to malate. Since malate to oxaloacetate was oxidation, this is what? This is reductive process. This is a reduction. Okay, reduction means, okay, something must be oxidized. Plants use NADPH more. So NADPH plus H plus, NADP plus oxidation happened. This is reduction again. Enzyme is your simple malate dehydrogenase, right? Malate dehydrogenase. Now malate, it passes or it goes through the mesophyll cell onto the bundle sheath cell via the plasmodesmata. They are the connections between the two cells in case of plants only. Desmatal connections are absent in animal cells. We have, you can say, the tight junctions, the gap junctions, occlutin, claudin, then integrin, cadherin, you can say, selectin and all. But plants have plasmodesmatal connections. Okay, so malate traverses from mesophyll cell to bundle sheath cell. Now you can see this is over. Now we have reached the bundle sheath cell. Okay. Now plasmodesmata. This one you can term it as plasmodesmata. Okay. We have reached, this is bundle sheath cell. This is your plasmodesmata. Okay, plasma membrane connections. Now malate has reached here. Now what you will see is a very drastic thing. Now malate will be oxidized to pyruvate. Okay, and this is not a simple oxidation. This is oxidative decarboxylation. Malate here is a 4 carbon compound, pyruvate is a 3 carbon compound. Always know the carbon numbers. That is why I had shown you the structures. I had drawn the structures for you while describing you or teaching you the TCA cycle slash Krebs cycle or glycolysis or EMP pathway, right? So, intern we have Pandas pathway, which is the most common pathway of glycolysis that is given in your syllabus. So, I have taught you that via drawing each and every structure. That is why, so that your carbon numbering is proper and you can comprehend it conceptually. The thing is, malate gets oxidatively decarboxylated to pyruvate. Okay, malate would become pyruvate. Enzyme is malic enzyme. Okay, this is what this is oxidative. You can write it down. It's 4 carbon. Okay. Now the first reaction here produces the first compound 4 carbon oxyl acetate, so it is known as the C4 cycle. Okay. This is 3 carbon. Yeah, it is written. So malate is 4 carbon, pyruvate is a 3 carbon molecule. Means one carbon went out, one CO2 out. So this is oxidative. One CO2 has been released, liberated, so oxidative decarboxylation. Just like we saw in case of the Krebs cycle where pyruvate underwent oxidative decarboxylation to produce acetyl CoA by the enzyme pyruvate dehydrogenase, which is a complex enzyme made up of E1, E2, E3. E1 was pyruvate dehydrogenase, E2 was dihydrolipoyl transacetylase, E3 was dihydrolipoyl dehydrogenase, right? And we needed various cofactors like NAD plus, FADH2, lipo8, right? Then you can say FAD, NAD plus, lipo8, and coenzyme A. Okay, so all those things and magnesium, we all needed all this. Now carbon dioxide is liberated. Now you have produced. This is what I told you is a, you can say, oxidative process. Oxidative means some reduction must happen. So NAD plus, which has come here, okay, from the mesophyll cell, NAD plus would be, then this NAD plus came in here in from the mesophyll to bundle sheath cell. Now that NAD plus, NADP plus will be reduced to NADPH plus H plus, right? Now since this is an oxidation, you need a side by side simultaneous reduction, because this is a redox reaction. You need to stabilize the lost electrons, right? So this is how you stabilize it and cause the perpetuation of a proper redox cycle. Now pyruvate would be then transported, will be traversing onto the, you can say, mesophyll cell again. Pyruvate will go back to the mesophyll cell, and then it will be converted to phosphoenol pyruvate. Okay, here the enzyme is a very special enzyme known as dikinase. Okay, dikinase, here ATP plus pi is converted to AMP plus PPI. Here ATP plus pi will be converted into AMP plus pyrophosphate is double phosphate, PPI. I'll explain to you how does this reaction happen. Why is it known as dikinase, not simple kinase? Just hold on. Now This reaction, first let me get down to the specificity. Why is it needed? This reaction, why did we go for this complex reaction? The answer to that question is, come to this one. Come to here. Now this CO2 that has been produced, okay, will be poured upon. Like you are standing here and I'm pouring, you can say a bucket of water on your head and you have nowhere else to go. You only have access to that water. So this, you can relate that analogy to the pathway here. The CO2, when the oxidative decarboxylation process is happening, the CO2 is being poured on, is being poured on to your, you can say, this thing, Rubisco. Okay, it is poured on to Rubisco and then it will catalyze the synthesis of ribulose 1,5-bisphosphate plus CO2. Here you have Rubisco. Now you can synthesize 2 into 3-phospho glycerate, right? Now just ponder and tell me, does this Rubisco have any chance of getting O2? Because you are not providing Rubisco any opportunity to select between O2 and CO2. If O2 is present, even though the Km is very high, almost 350 micromolar, then it can even go for 3:1 ratio because O2 concentration is high, almost 20%, CO2 is 0.04% currently. Then it can go for that reaction. But what I am doing is I have only made available Rubisco in the bundle sheet cell. I am not making Rubisco available here in the mesophyll cell so that it can take either O2 or CO2 from the air. So I'm only making Rubisco express in the bundle sheath cell. And in the bundle sheath cell, only CO2 is being released. There is very low or minuscule probability that it will go for oxygenase activity. So there is no or negligible photorespiration. Just try to think and contemplate how, you can say, witty, how great the nature is, right? How extraordinary the evolution process is. It has almost eliminated, okay, almost it has completely eliminated the oxygenase activity, not by mutating the enzyme, not by doing any kind of, or not by committing any kind of mutation in the enzyme in the protein structure, but by restricting oxygen inflow or influx into the cell and by restricting the Rubisco expression in the, or by confining the Rubisco expression in the bundle sheath cell only. So that is how it has been able to restrict or prohibit the, you can say, the oxygenase activity of Rubisco. Okay, so the thing is, this is how it works. Okay, this is how this thing occurs, and nature has been great in applying this and achieving this evolutionary success. Okay, now coming on to the dikinase reaction. The pyruvate formed by decarboxylation of malate in the abundances cells is transferred back to the mesophyll cells where it is converted to phosphoenol pyruvate again by a very unusual enzymatic reaction catalyzed by a dikinase enzyme known as pyruvate phosphate dikinase, or simply a dikinase. Okay, now this is called a dikinase, okay, because the book doesn't give you this explanation. Just writes, some books have written kinase only. They have made such blunder that they have made it written kinase. Some books have written dikinase, but the explanation is absent. Dikinase, I don't know, two times kinase. It is not like that. Okay, this enzyme is referred to as a dikinase. Why? Because two different molecules are simultaneously phosphorylated by one molecule of ATP. Okay, two phosphorylation. One phosphorylation event means one or solo or solitary kinase activity, and the donor of the phosphate group is ATP. But if two is being phosphorylated, again donor is ATP, it means double kinase activity, so it's a dikinase. Number one is pyruvate to, you can say, PEP, okay, and phosphate to pyrophosphate, okay. So first of all, pyruvate to PEP happens, okay. So pyruvate is phosphorylated to become PEP, and phosphate is phosphorylated, okay, to become pyrophosphate. If you see this reaction, okay, just look at this reaction. This one, this pi is added extra, so this phosphate becomes pyrophosphate, okay, and one of those phosphates, AMP will release two PIs, okay. Don't get confused here. AMP has released, ATP has three phosphates. You will say AMP already has released two PI, but one PI, okay, one phosphate of from this has went or has gone on to phosphorylate pyruvate to form PEP. Now one phosphate remained. This phosphate again got phosphorylated by, you can say, another phosphate. Okay, another phosphate coming in, and plus pi, this has again become a pyrophosphate. Okay, so diphosphate. One has phosphorylated the pyruvate, one has phosphorylated the phosphate. So you can say here that if you are writing dikinase, one is pyruvate, two phosphoenol pyruvate, okay, or PEP. Another one is phosphate to pyrophosphate, means Pi to PPI. Okay, phosphate to pyrophosphate, and then the pyrophosphate is subsequently hydrolyzed to phosphate. So two high energy phosphate groups of ATP are used in regenerating the phosphoenol pyruvate. Okay, and then the phosphoenol pyruvate is now ready to receive another molecule of CO2 in the mesophyll cell. And one more thing is very, very important, that why did we convert this CO2 to SCO3 minus? Okay, the PEP carboxylase enzyme of mesophyll cells has a high affinity for HCO3 minus, okay, which is heavily favored relative to CO2 in aqueous solution, okay, and can fix more CO2. Point number two, compared to Rubisco. So I told you the turnover number of Rubisco is very, very low. That is, its concentration is very, very high. But this can accept more SCO3, not CO2, more SCO3 than, you can say, compared to CO2. Okay, and if CO2 entered, okay, then CO2 could have directly diffused, okay, to the cells, to the bundle sheath cell, and then O2 could have also diffused. So in order to make this dramatic, you can say, change, it requires HCO3 minus, not CO2. Okay, so it has blocked the CO2 entry also in order to be on the safer side. Okay, and it is very, very highly, the PEP carboxylase has high affinity for HCO3 minus compared to CO2. Okay. Another way of analyzing this entire equation. Okay. So the PEP carboxylation reaction then serves to fix and concentrate CO2 in the form of malate. Okay. So unlike Rubisco, it does not use O2 also. The PEP carboxylase does not use O2 as an alternative. substrate. So again, there is no competition between CO2 and O2 again. So first of all, this one, and does not use O2 and does not need to increase its enzyme concentration. That is why it is much more effective, means turnover number is much higher than Rubisco. That is why again it uses HCO3 minus. Okay. So much consideration the nature has taken, using SCO3 minus as the substrate, then using, you can say, the CO2 as the decarboxylating, you can say, consequence, or the consequence of oxidative decarboxylation, and only causing the expression of Rubisco in the bundle seed cell only, not in the mesophyll cell. So those things play a dramatic role, okay, in the perpetuation of Krebs cycle. Now, once CO2 is fixed into 3-phosphoglycerate in the bundle seed cells, the other reactions of the Calvin cycle can take place exactly as I have described earlier. Okay, so thus, to summarize, the C4 plants, in C4 plants, mesophyll cells carry out CO2 assimilation by the C4 pathway, and bundle seed cells synthesize starch and sucrose by the C3 pathway. Okay, so that is what you should be knowing. And three enzymes of the C4 pathway are also regulated by light, okay, becoming more active in daylight, like malate dehydrogenase. But those specific mechanisms of thioredoxin dependent reduction is not in your course, so we will not go into that detail. That happens in the C3 cycle also. That is why we skipped that detail in C3 cycle as well. Okay. Now the last topic of this chapter is the CAM plant. Okay. So CAM plants means cressolation acid metabolism. Now succulent plants, okay, CAM pathway. Now succulent plants, such as, you can say, cactus, pineapple, which are native to very hot and extremely dry environments, have another variation on photosynthetic CO2 fixation because they cannot lose water. So this will reduce their loss of water via water vapor through pores, that is stomata perpetuated by guard cell opening, okay, by which CO2 and O2 must enter leaf tissue. So what they do is, so what they do is, instead of separating the initial trapping of CO2 and its fixation by Rubisco across space, like it is done in the C4 plants, like mesophyll and bundle sheath cell, they are separating it across space into two different cell types. So instead of separating, okay, the initial trapping of CO2 and its fixation by Rubisco across space, okay, like what happens in the C4 plants, they separate these two events over time. Very, very important. They separate these two events over time. So time function, okay, not the space function. Now what do I mean is at night when the air is cooler and moisture, the stomata open to allow entry of CO2. Okay, at night stomata open, CO2 enters. Okay, CO2 enters. Now, when CO2 enters, it is then fixed into oxaloacetate by the same enzyme PEP carboxylase. Okay, here when the CO2 enters, it is fixed to oxaloacetate by the same enzyme PEP carboxylase. But the oxaloacetate again will be reduced to malate. Oxaloacetate is reduced to malate, same reaction as we saw in the C4 cycle. But now the difference will come in. Now the malate is stored in the vacuoles. And why stored in the vacuoles? Because malate is malic acid. So if it is not stored in the vacuoles, what it will do is it will increase the acidity, right? It will increase the acidity of the cytosol, and therefore in order to protect the cytosolic and plastid enzymes from the low pH produced by malic acid dissociation, it is stored in the vacuoles. Okay, that is the reason. Now during the day, the stomata they close and prevents the water loss that would result from high daytime temperature, and the CO2 which has been trapped overnight in malate is now released as CO2 by the NADP linked malic enzyme pathway that I had just shown you. Now this CO2 is now assimilated by the action of Rubisco and the Calvin cycle enzymes, because in the daytime the entire stomata is shut down, so no CO2 can come in. Okay, again the competition is stopped between CO2 and O2 because nothing can come in, and at that point in time, okay, the CO2 will be released by the same pathway malic acid enzyme, and then the process will go on. So night and day it has been differentiated. So at daytime the starch synthesis will happen, and that will help it at the day and night both. Okay, so that is how the CAM plants have been able to generate, okay, the starch. And since this method of CO2 fixation was first discovered in stone crops like perennial flowering plants of the family Crassulaceae, now this is known as Crassulacean acid metabolism. Okay, Crassulacean acid metabolism, that is CAM plants. Okay, then the plants are known as CAM plants. So I hope that you were able to comprehend the lecture. If you had any kind of doubts or queries, you can definitely ask me on my personal number to resolve your queries. Okay, then thanks a lot.